Hello everyone, I'm Zhibin from BetaScience. I'd like to walk you through our new flagship model in this video, the BeNano 180 Zeta Max, and introduce its 3 technical breakthroughs and innovative features. But before we dive into the details, let me take you back to where the journey began. Our story started in 2012, when BetaScience launched its first generation nanoparticle analyzers, the non-optic 90 for particle size, and the BD-Z100 for zeta potential. That marked our official entry into nanoparticle characterization. After years of building up experience, in 2020, we released the BeNano series. This new generation integrated advanced techniques such as pulse, backsketching detection, microreology, and DLS flow mode. This made BeNano more versatile and suitable for a much wider range of applications. Since then, our R&D team has continued to move forward. The team kept breaking through challenges and developed new features in-house. This included refractive index measurement, particle concentration, sedimentation-based sizing, and transmittance measurement. All of this paved the way for the BeNano 180 Zeta Max. The key advantage of the BeNano 180 Zeta Max is that we integrate multiple techniques into a single compact system. It has 14 measurement modes. It includes DLS for size, microreology, VVVH polarizer, fluorescence filter, and flow mode. ELS for zeta potential, automatic pH titration, and temperature trend. SLS for molecular weight and distribution. And on top of that, patented techniques extend the BeNano to refractive index measurement, particle concentration, and even micron size analysis through sedimentation and transmittance measurement. With all these functions working together, BeNano covers samples from nanometers to micrometers, and from dilute solutions to concentrated systems. It delivers high-precision analysis of size, surface charge, concentration, and molecular weight, meeting the needs of both scientific research and industrial applications. With dynamic light sketching DLS, the BeNano 180 Zeta Max offers measurements of the average size, polydispersity index (PDI), and particle size distributions by intensity, number, and volume. It also determines the diffusion interaction parameter (KD) and provides insight into rheological behavior. Through phase analysis light sketching (pulse), it delivers average zeta potential change as a function of pH, and the isoelectric point. Using laser Doppler electrophoresis (LDE), it characterizes the zeta potential distribution. With static light sketching (SLS), it determines absolute molecular weight and the second virial coefficient. Beyond this, the system integrates light extinction (DLS), LEDLS, to quantify the volume fraction and particle number concentration of each population. Sedimentation particle sizing further enable parameters such as D10, D50, D90, span, volume-weighted mean diameter, and detailed size distributions by volume. With optical refraction, it identifies the dispersant refractive index, while transmission determination provides the average sample transmittance. Altogether, these parameters highlight the comprehensive measurement portfolio of the BeNano 180 Zeta Max. BeNano meets many regulatory requirements. It complies with ASTM, ISO, and the US Pharmacopoeia. The system offers instrument qualification, including IQ and OQ. The software supports FDA 21 CFR Part 11. This ensures secure data, electronic signatures, and audit trails. These features make BeNano suitable for pharmaceutical companies and other regulated industries. What truly sets the Max apart from other DLS analyzers is the integration of the RCS module. The RCS module is positioned at 0 degrees and it combines a photodiode with the CMOS camera. This setup allows the system to measure not only refractive index and particle concentration, but also micron particle size through sedimentation. These are the features that I will explain in more detail in this talk, and this makes the BeNano the most comprehensive models in the series. Let me now show you how these novel functions work in practice, starting with the refractive index measurement. The BeNano 180 Zeta Max includes a liquid R test based on a patented wedge-shaped cuvette. A laser passes through the liquid, and a CMOS camera records the shift of the light spot to calculate R. Calibration is simple. Just use two reference liquids such as water and DMSO, build a linear curve, and you are ready to measure. In real testing, BeNano was used to measure the R of 6 samples, with the results closely matching the nominal values. Deviations were extremely small, absolute deviation below 0.001, relative deviation within 0.1%. The results showed a stable linear relationship, proving accuracy across the different sample types. This feature supports R values from 1.2 to 1.6, making it especially useful for dispersants with unknown R values in DLS or ELS, whether water-based or organic. Moving on to another key feature, the BeNano 180 Zeta Max can also run a particle concentration test using our patented technique. With this approach, BeNano can determine both the volume fraction and the number concentration of particles. The testing process is straightforward. First, in light extension measurement, the laser passes through the blank dispersant and the sample, and the forward PD detector records the transmitted intensities I0 and IT. Second, a DLS size test, either the 90 degrees or 173 degrees, is selected to obtain the size distribution and calculate D32. Then, combining these results with mean theory, the extension coefficient QE is calculated. When all the parameters are put into the equation, the overall volume fraction phi is obtained. Once phi is determined, we can go a step further. By multiplying phi with the volume percentage of each peak, we get the volume fraction of each population. Then, using the mean size of each peak, we can relate volume to size and calculate the number concentration of each particle group. That's how this feature works. To validate this method, BeNano was used to measure different polystyrene standards. Results match the theoretical values very well. As shown in the figure, BeNano can measure 20 nanometers to 500 nanometers, and concentrations from 10 to the power of 7 up to 10 to the power of 16 particles per milliliter, covering most nanoparticle studies. The concentration measurement of the BeNano is easy to operate, fast to perform, and particularly useful for sample screening tests. It works for both aqueous and organic systems and plays a significant role in quantitative analysis of suspensions and process monitoring. Let me give you a real-world example from the semiconductor industry. Silica-based CMP slurry is a very important material, and its process stability is strongly affected by concentration. Using BeNano to test silica at different dilution levels, the size distribution curves were always single peaks, proving that the sample had excellent monodispersity. Looking further at the concentration results, the dilution factor is 10, and we find a clear tenfold linear relationship between the measured concentration and the dilution factor. The deviation compared with the theoretical value was less than 1.5x, which confirms BeNano's accuracy and stability in real-world applications. Through tests like this, users quickly check whether their sample concentration is reasonable or not, providing strong data support for both R&D and production processes. In addition to nanoparticles, BeNano is also capable of measuring larger particles with a sedimentation-based sizing function. This feature is designed for nanomaterial systems that contain micro-sized particles, which are often beyond the reach of traditional DLS. Here's how it works. A laser passes through the sample, and the PD detector at 0 degrees continuously records the transmitted intensity. As time goes on, particles gradually settle under gravity and the transmission increases. Larger particles settle faster, while smaller ones settle more slowly. By analyzing the transmitted intensity versus time curve, BeNano can calculate the settling velocity. Then, applying Stokes' law and knowing the dispersant viscosity as well as the densities of particles and the dispersant, the BeNano calculates the particle size, also known as the Stokes equivalent diameter. The upper limit of the measurement can reach 50 micrometers, making it a powerful complement to conventional DLS for larger particle detection. Besides that, BeNano was used to measure PMMA samples with particle sizes of 5, 10, 15, and 20 micrometers. The samples were mixed in different combinations, and the sedimentation method successfully measured them with very high size resolution. It was even able to distinguish mixtures where the size ratio was as close as 1 to 1.5. At this last part of the presentation, I'd like to share some real-world applications of BeNano with you. BeNano has already been cited in top international journals, including Nature Communications, Science Advances, and Advanced Functional Materials. This shows how widely it has been recognized in cutting-edge research. One example comes from the Journal of Controlled Release, a leading journal in drug delivery. The researchers created a special manganese oxide nanostructure using a short peptide as a template. To check its properties, they used a combination of AFM, TEM, and BeNano. The results matched perfectly, confirming the particles were uniform, nano-sized, and stable. This case really shows how BeNano works hand in hand with other techniques to give solid, reliable data. Another example was published in 2024 in Nature Communications. The study focused on new single-atom nanozymes. The team used BeNano to measure surface charge and compared different materials. The results revealed how molecules interact and form stable structures, giving insights into the link between surface properties and material performance. BeNano played a key role in uncovering these details. In a separate study, researchers worked on fullerene derivatives, also known as buckyballs. They wanted to see how chemical modifications affect stability. With BeNano, they measured zeta potential and confirmed that one modified form was much more stable because of the way it interacted with water molecules. This finding supported their design strategy and helped explain why the particles behave differently. Finally, in the field of genetic diagnostics, BeNano was used in a study reported in the Journal of Nanobiotechnology. The researchers developed a dual nanoparticle system for RNA detection. BeNano confirmed that the particles were uniform, well dispersed, and carried the right charges for stability. These results gave the team confidence that their nanosystem could serve as a reliable platform for gene detection. All of these cases highlight BeNano's versatility and its value across very different research areas, from drug delivery to nanozymes to advanced biomaterials and genetic diagnostics. And as I have shown earlier, with the newly added functions such as refractive index, particle concentration, and sedimentation-based sizing, BeNano 180 Zeta Max is not only versatile but also ready for the future needs of researchers. That brings me to the end of the presentation. Thank you all for your attention.